Hello everyone. Today we are going to talk about GAT B 2021, which is a major biotechnology entrance exam if you are preparing for MSc. And in these exams, there are few new topics like cell culture, organ culture, and tissue culture, which we can discuss in this video. In this video, we would focus on the topics that you need to learn, and secondly, we would practice 5 questions, which would range only 10 minutes of your time. So stay tuned till the end of this video. Now, GAT B 2021 or 2020 had all those classic syllabus that it had previously when it was JNU CEB, like microbiology, cell biology, molecular biology, fundamentals of biochemistry, immuno, genetics, and methods in biology. All of these things were there before and they are still there now. But there are a few new additions, and people are going nuts about how to prepare for these particular topics. These new topics include bioinformatics, bioengineering and bioprocessing, animal and plant tissue culture. Now, if you want to learn more about these things, I have a crash course on that. You can always go join an academy using my code AP10. You'd get a 10% discount. But let me tell you more about these things in this video. If you're thinking that how to prepare for these parts, how to clear these concepts, and you don't have a clear concept or idea about these parts, an academy subscription could really help you, because there is a dedicated batch known as Synapse batch, which is starting from 28th April 2021. It would guide you for all these new topics. They would help you to cover all of these new things. At the same time, you can practice a lot of questions. But the price would rise very soon, so 5 days left for that. Subscribe right now. In this pandemic, study safe and study from your home. Your unacademy educators are always at your fingertip. And lastly, download the app, use my code AP10, then you can start solving questions, you can start learning from the educators, clear your doubt with them, and many more. So enroll right now. Let me tell you the topics that are really important and that need to be discussed. These are animal and plant tissue culture basics, organ culture for plant and animal, cell culture methods, cell culture media, hybridoma technology, stem cell technology, and transgenic animal generation. So these are important topics that you should know before going to the exam. Now, in this particular video, it's not possible to talk about all of these things, but I would list all of these concept-bearing videos in the I button. So click on the I button for more information. Let's talk about question number 1. In question number 1, we would talk about a particular question where we see that name the type of culture which is prepared by inoculating directly from the tissue of an organism to the culture media. This is known as primary culture, secondary culture, or cell lines, or transformed cell culture. So let me tell you, the answer would be primary culture. Now, if you want to know more about primary culture or what are secondary cultures, what are cell lines, then you should go and read more about these things in my separate video. So the video link is in the I button. So you can go there anytime. So these terminologies, they might ask. They might not go really deep into these topics because these are all practical, methodological things. So you would eventually learn them. But you just need to have an overview of these processes. Similarly, this question is another example. That what is a cell line? Multi-layered culture, transformed cell, multiple growth of cells, subculturing from primary culture. So it turns out the option D is correct. Now, if you want to know more about it, you have to watch the video which is in the I button. Okay. Then there is a question which says which of the following is an characteristic of normal cell? Encourage independent, continuous cell line, dependent on external growth factor, no contact inhibition. So let me tell you, the correct option here would be dependent on external growth factors, because other characteristics such as continuous cell line, no contact inhibition, anchorage independentness, these are characteristic of cancer cell line, not a normal cell line. So option C would be correct. Okay, question 4 says why subculture of cell is necessary. First, one statement says not enough space, so the T is omitted here, but anyway, not enough space for the cell to grow. Accumulation of toxins, not enough nutrients for cell growth. And which would be the correct option? All of these above are correct options. Now you want to know that what are subculturing technique or what is a subculturing technique? You can learn about it in the I button. Okay. Then the question number 5 says the hybridomas are made by fusing T cells with myeloma cells, fusing B cells with myeloma cells, fusing T helper cells with myeloma cells, fusing memory B cells with myeloma cells. I vaguely remember that you guys read about hybridoma technique in your class 11 and 12, so you might know this answer. If you know this answer, comment in the video and let me know which would be the correct option. And if you don't know the answer, it's completely okay. You can learn about hybridoma technique in the video which is given in the I button, learn from there, and then try to answer this question. Okay, if you like this video, give it a quick thumbs up, and don't forget to like, share, and subscribe. Thank you. As usual, please let me know in the comment how you like this video. Thank you guys.